This recorded session is dedicated to our discussion of um, key examples of endospore forming bacilli, which are medically significant. So we will focus on two genera, Bacillus and Clostridium. Bacillus are a genus of spore-forming bacteria that are either or that could be aerobic or facultatively anaerobic. And most of them are found in the environment, particularly in soil and water. And a key feature of this group is the formation of endospores. Endospores are survival structures, meaning they allow for prolonged prolonged presence of the species or the genus in different reservoirs and sources. Endospores are cellular structures which are resistant to drying, radiation, to low pH, and to toxic chemicals. So they're not really cellular per se, but they are structures that contain a complete set of genetic material allowing for propagation or activation of um, um, actually not activation but more of propagation of the genus once there are stress signaled or stress perceived from the environmental or from the ambient. No? Endospores can be dormant for really long periods of time. Okay. So now let's look at some examples of Bacillus species that are clinically significant. So first is Bacillus anthracis, which is the causative agent of anthrax, usually affecting herbivore animals. So morphologically, the species appear as blunt-ended bacilli, so you can see in this photo micrograph. Highlight. Okay, so it's blunt-ended, and the spores are located central to the bacterial cell and are appearing as oval-shaped structures. They are non-motile and they do not really release hemolytic enzymes. When grown on blood agar, they don't show hemolysis. The capsule of Bacillus anthracis is one virulence determinant. It's unique as the component is only made up of, or the structure itself, the entire capsule is only made up with poly-D-glutamic acid. So it's unique to Bacillus anthracis. So it's a good marker for the detection of this pathogen. Okay. Now, the major virulence factor of Bacillus anthracis is actually a set of released exotoxins. So, B. anthracis produces a tripartite or three-component exotoxin, an edema factor, a lethal factor, and a PA, which stands for protective antigen. So there are these three. Individually, they are not toxic. They're not able to um, uh, elicit pleiotropic response from the host, but together they are toxic and they produce systemic effects of the anthrax infection or the anthrax disease. No? So let's take a look at these three toxins produced by B. anthracis. So first is the edema factor. So EF, it's an extracellular form of a cyclase, an adenylate cyclase. which allows the increase in intracellular cyclic AMP. And the toxin, this component of the toxin, serves as an anti-phagocytic factor. PA, protective antigen, um, promotes entry of the edema factor into the phagocytic cells. So this protective antigen binds to host receptors, then allowing for translocation of the edema factor toxin. LF, lethal factor, causes pulmonary edema and death in the animal model rats. They inhibit, or the toxin inhibits particular signal transduction pathways that contribute to apoptosis, to contribute to host death, no, to host cell death. The edema toxin is primarily composed of the edema factor and the protective antigen. And on the other hand, lethal toxin is composed of the lethal factor and the protective antigen. Okay? So there are different forms of anthrax, and the three general ones, or the three well documented ones, are cutaneous anthrax, respiratory anthrax, and gastrointestinal anthrax. There's a newer type of anthrax identified in a particular demograph called welder's anthrax, or metal workers anthrax. So this fourth type has been identified in metal workers, in welders. It's associated with pneumonia production, and usually it's fatal. No? So for the three other forms, we'll talk about them. So first is cutaneous. Cutaneous anthrax is the most common example or manifestation of anthrax. If the route of entry of the pathogen is via cuts or injuries in the skin. On the other hand, respiratory anthrax, or wool sorter's disease, is also called pulmonary or inhalation anthrax. Gastrointestinal anthrax, as the name suggests, affects gastrointestinal organs. So here, let's take a look at each. So cutaneous anthrax, you can see the manifestations are eschar formation, black eschar formation, usually on the neck, on the face, on the limbs, or particularly the arm portion. Respiratory anthrax inhalation affecting the lungs, and gastrointestinal, on the other hand, affects the oral cavity down to the GIT organs. Anthrax is actually preventable. There is vaccine available, especially advisable for high risk individuals, those individuals with frequent contact with the environment, with those that work with soil, those that might get the spores of the bacteria, or the bacterium itself, from as part of their occupation. So those with occupational hazards, no. The treatment for this pathogen is penicillin G and erythromycin. Okay. Then we have another Bacillus species. We have B. cereus, which is motile and does not produce capsule. It is resistant to penicillin, and it can cause two types of intestinal disorders depending on the toxin that is released. No, so two types of enterotoxins associated with gastro diseases are the labile toxin and the heat labile toxin and the heat stable toxin. Heat labile toxin of B. cereus is associated with diarrheal disease. So there's diarrhea manifesting. And heat stable, on the other hand, is associated with an emetic disease. So Emetic meaning causes vomiting, no? So, B. cereus is a foodborne pathogen. It is acquired, or it can gain entry to the host animal, to humans via food. Now, another group that we will emphasize in this recorded session are the Clostridia, the Clostridium, which are obligately anaerobic, do not require oxygen, so their culture conditions are primarily anoxic, and they appear as blunt end rods, blunt end bacilli. Most of the species of Clostridium are motile, and similar with bacilli or bacillus, they're found in soil, in aquatic sediments, in the environment. Their spores frequently contaminate produce such as vegetables, those that are practicing home gardening. Okay, so there's high risk of acquiring Clostridium spores or the bacterium itself from the produce. Also associated with meat products such as smoked fish and also fresh honey. Clostridium species secrete very powerful exotoxins and enzymes that contribute to their overall virulence. Okay, so here is, let's look at specific species. So we have Clostridium botulinum. Let's examine. Let's discuss a few about the medical significance of C. botulinum. It's a causative agent of botulism, which is a rapidly manifesting type of food poisoning. It produces extremely lethal neurotoxin, the botulinum toxin, with the mechanism of action where it blocks the release of acetylcholine from presynaptic nerve terminals in the ANS and motor end plates, preventing or blocking proper muscle contraction. So it causes muscle paralysis. Oops. Okay. Now, the kinds of diseases that C. botulinum infection may cause include adult botulism, which is associated with consuming smoked fish or home canned vegetables or products. Adult botulism can cause systemic shock, which is usually afebrile without fever. There are manifestations such as diplopia, dysphagia, the difficulty in swallowing, muscle weakness, and paralysis of the respiratory organs. So there is also an effect on the respiratory system, or the lungs. Okay. Also, there is infant botulism. Okay. Uh, Also, what do you call this? Associated with the term floppy baby syndrome, could be the result of ingesting contaminated honey. So some of the symptoms include constipation, dysphagia, and muscle weakness. Okay. Then we also have another species of Clostridia, Clostridium tetani, which is the causative agent of tetanus, as the common route or common event allowing the introduction of the species to a human host is punctured wounds, especially on the foot area. Okay, cellularly, the species appears as long, slender rods with a round terminal endospore. Okay, they secrete an exotoxin, a powerful neurotoxin, the tetanospasmin, which supports or causes sustained contraction of skeletal muscles. So the skeletal muscles cannot relax. No, if with in tetanus diagnosis, the skeletal muscles keep on contracting. Okay, so here it's the appearance of C. tetani. So you can see the terminal endospores. Okay, so you're seeing some pictures as visuals of the symptoms. Let's take a look at symptoms. So there is severe muscle spasm, especially in the muscles of the jaw. Okay. There's also spasm of facial muscles, also called risus sardonicus. So you can see the grinning expression as manifested or as shown in this visual. Risus sardonicus. And there's also opistothonos or noose, which is abnormal arching of the back, manifested by some patients with tetanus infection. So here, opistothonos, you can see the arching of the back. Okay. The mortality rate is very high once that there is already lockjaw for the case of tetanus patients. How to prevent it? There is a antitoxin that can be used, the tetanus toxoid antitoxin. Okay. Then there is Clostridium perfringens, which is the common causative agent of gas gangrene. So gas gangrene is characterized by the death of soft tissue on the site of infection due to loss of blood supply and associated gas production. The species is encapsulated, it's non-motile, and C. perfringens is actually very widely distributed. They're ubiquitous in nature. Okay, they're part of our normal flora in our GIT and in the vagina for the female humans. Here you can see um, C. perfringens. Okay. Then the toxins, for the toxins formed, so C. perfringens can secrete exotoxins and enterotoxins. So exotoxins, there are about 12. And the most important one is an alpha toxin, which is a lecithinase. It is targeting lecithin in the cell membrane. An enterotoxin of of Clostridium perfringens affecting the host gut cells. They are secreted upon invasion of the host gut cells, um, causing imbalance or disbalance in ion transport of ileum mucosal cells. They also allow, or this toxin also disintegrates tight junctions between epithelial cells of the gut. So really destroying the structural integrity and cellular balance, no? Intracellular balance of the host cells. Okay. C. perfringens in relation to its very important sequelae, which is destruction of soft tissue, primarily attributes that capacity with its degradative enzymes. So it's able to secrete hydrolytic enzymes, collagenases, hyaluronidases, proteases, and DNases, which are all responsible for tissue liquefaction and allowing for further invasion or spread of the infection. Okay. Oops, what's happened? Okay. Now, some of the diseases caused by C. perfringens infection. So we have Clostridial myonecrosis, so Clostridial gas gangrene, characterized by death and decay of wound tissue due to deficiency and cessation of blood supply. So there's putrefaction with gas, so there's production of sugar, there's sugar fermentation in the tissue. So the sugars that are released during the destruction of soft tissue are fermented, providing an anaerobic environment, allowing for C. perfringens cells to continue dividing or continue propagating. It's happening. Okay, wait. And then, okay, so here you can see gas gangrene, clostridial gas gangrene. Okay. Oops, what's happening? Okay. More on Clostridial myonecrosis. So there's, okay, so I mentioned all of this already. Okay, yeah. So in cases of wide distribution, or in cases when the pathogen has become more systemic because of, let's say, for example, a wide portion of tissue already affected and causing systemic gangrene, the organisms can progressively destroy tissues proximal to the site of infection and invasion. And that can lead to septicemia, can lead to a more toxic situation where um, the blood supply has already been invaded and the pathogen can be distributed in other parts of the human host. Okay? Okay, so I mentioned this already. Hmm. Okay. Aside from gas gangrene, from clostridial myonecrosis, there are also other clinical manifestations of C. perfringens infection, such as cellulitis, okay, especially if it's affecting or if the pathogen gains entry to the host cell via a wound infection or an injury of the skin on the skin. Then food poisoning, definitely, associated with spores of the species. Then there is enteritis necroticans, which can cause or which is characterized by death of intestinal tissue. For females, there is clostridial endometritis, the inflammation of the endometrium portion of the the female the female reproductive structure, no? The primary female reproductive organ, the uterus. Okay. Primarily what's being affected are the mucous membrane lining the uterus for clostridial endometritis. Okay. So how is it treated? How is C. perfringens treated? So there are several options. First is radical surgery, meaning the portion that is affected will be excised, will be amputated. So it's common that in very in worst cases of C. perfringens infection, the affected tissue, the affected body part is removed. How to treat it? There are usual antibiotics that can be prescribed, such as penicillin and clindamycin. And in some cases, hyperbaric oxygen chambers are used. So in this example or in this treatment approach, oxygen is saturating the tissue, so killing the pathogen, killing the bacterial cells. I don't know what's happening. The slides keep on going back to slide 23. Anyway, all right, and then we have C. difficile, which is the agent of pseudomembranous enterocolitis. C. difficile is part of our normal flora. About 2 to 3% of healthy individuals globally contain it or has it as a normal flora. It's transmitted via the orophycal route, and virulence determinants of this species are toxins produced by the cell by the pathogen. Toxin A, which is an enterotoxin capable of causing diarrhea, and toxin B, which is a cytotoxin toxic to the host cells, to colon cells. Okay, so here is a visual to help us have a more concrete understanding of the a more concrete visualization of the clinical manifestations of C. difficile infection. So C. difficile responsible for antibiotic-associated pseudomembranous enterocolitis. So you can see here, inflamed mucosa with white exudates or with areas of white exudates, which are the pseudomembranes on the surface of the colon tissue. Okay? So what happens or the usual route is if a person undergoes antibiotic treatment, especially for the case of broad spectrum antibiotic regimen, such as ampicillins or clindamycins, C. difficile can dominate the gut microbial communities and can become very pathogenic or can be virulent depending on its genetic makeup. If it's able to secrete those toxins, A and B, then it can cause the clinical manifestations of enterocolitis, of pseudomembranous enterocolitis. And recently, or I mean, in the contemporary times, drug-resistant C. difficile strains have been identified. So it's actually not emerging, but it's I think C. difficile is already one of the um, uh, highly monitored bacterial pathogens causing human infection. Okay, so. Uh, Okay. So how is C. difficile infection treated? So uh, we of course we can definitely there's consideration of stopping the use of antibiotics if it's the causative agent of the dominance of C. difficile or the prevalence of C. difficile in the gut. Oral metronidazole is also used. Metronidazole is actually an antiprotozoal, but can be effective in treating or countering the effect of anaerobic infections. And also there is vancomycin as an antibacterial. Okay, so that ends our discussion for endospore forming uh, bacilli. So the last, I'll be recording another one, the last handout on bacillus, on non-spore forming ones.